Welcome, everyone. My name is Daniel Giraldi. I'm Associate Director of Technology Markets here at Oxford Nanopore. I'm really excited to welcome you to this webinar, this knowledge exchange, where we're going to talk about the latest updates to direct RNA sequencing from Oxford Nanopore. After the talk, we will have a live Q&A session. So please add your questions to the Q&A box, which you can find at the bottom of your screen. You can also upvote questions from other people if you would like those to be answered at the top. The knowledge exchange today is being recorded and will be made available after the broadcast on demand. I'd now like to welcome Libby Snell. Libby is Director of RNA and cDNA Sample Technology at Oxford Nanopore. And Libby and I worked together on the first version of our direct RNA kit, which we published that work in Nature Methods in 2018. And Libby and the team have done a lot of work since then, and you've probably seen a recent update from them. We're really excited to hear today from Libby about the latest version of the kit.
Hello, my name is Libby Snell, and I am the Director of RNA and cDNA Sample Technology here at Oxford Nanopore. I have been with the company over 12 and a half years, and I am one of the original lead designers and developers of direct RNA sequencing, working on this since its very beginning. So I am very happy to sort of walk you through some of the attributes of direct RNA sequencing and how it can be used to sort of enhance your own research. So by way of introduction, I'm sort of going to give you a background a bit of RNA sequencing in general, and then sort of move into why direct RNA sequencing is such a good fit for a lot of different questions that you might have. So when it comes to RNA sequencing, obviously it was a significant breakthrough for many different applications. It is usually one of the most interesting, I think, methods of looking at a particular research question, because it gives you dynamic information of what's happening in cells or tissues at any given time. And it's sort of a revolutionary advancement with different applications. So things like biomedical research, so when looking for biomarkers for particular diseases, and looking for the underlying molecular mechanisms of disease. We can look at drug and vaccine development. This is obviously quite relevant recently in the last 5 years. And then also things like agricultural, so looking at improving crop quality, optimizing plant breeding, working on ways of sort of enhancing the genetic engineering that we would see to sort of help mitigate things like availability of crops for high salinity or drought resistance and things like that. So it's one of the things that people tend to look at when you're trying to figure out what is actually happening within an organism, as opposed to just what is the information that's available, such as when you're doing genetic sequencing of a genome. So traditionally, or the legacy way of looking at cDNA or direct RNA, RNA sequencing is through changing RNA to cDNA. In the past 40 odd years or longer, maybe 50, people have been have had the ability to sequence RNA, but you've had to convert it to cDNA. So the process would be RNA isolation, then you convert it to cDNA, and then you do your library prep, you do PCR usually, not always, and then you sequence and you do your data analyses. And this is very good. It's still very useful. However, there are some things that you kind of lose as far as the information when you're doing cDNA type RNA sequencing. So you get loss of base modification information, or RNA modifications. They can be either on the nitrogenous base itself or on the sugar backbone. We also lose in traditional ways of looking at this, or legacy ways of looking at this, we lose poly-A tail length insights. So poly-A tail lengths are very important when you're looking at sort of transcript life cycles. We know that as transcripts are either being transported out of a nucleus into the cytoplasm where it's going to be translated, we lose some of the poly-A tail length. Transcripts that are going into storage tend to be very short and then get repolyadenylated. So this kind of, the ability to be able to look at poly-A tail lengths is very important if you want to find out exactly what's happening to your RNA transcripts that you are engaging with as sort of at the ribosomal level. And the other thing about cDNA conversion is sometimes we lose isoforms because they don't map very well because of flaky reverse transcriptases that don't necessarily get to the 5' end, and I'll explain this a bit later, or PCR. Also, you get PCR biases, so you get inaccurate expression quantitation. So if you want to look at what genes are actually being transcribed and therefore the levels that they're being transcribed, if you have PCR, sometimes certain transcripts that are shorter will get overamplified, and therefore you get an inaccurate representation of what might be there. And also some PCR, some transcripts don't PCR very well just in general because they have maybe a repeat sequence or something like that. So it kind of changes the actual information that you're trying to look at. It's also very expensive. It can be expensive. It can be very time consuming depending on the prep that you're doing. And then you also get sort of a long time to wait for real-time results if you're using specific platforms as far as sequencing. So while it's good, it's not always full information that, would sort of reveal that you might want to know from your particular RNA sample. So what I think is a better way of doing it is direct RNA, which is using a cDNA-free type of RNA sequencing, which reveals more biology, and therefore you get a better look at actually what's going on, that dynamic information that's going on in the cell at any given time. So we see things like RNA modifications, so we can detect those. We get highly accurate poly-A tail insights, which means that we can measure the poly-A tail lengths, which, as I mentioned before, can give you an idea of what's actually happening to those transcripts at any given point. We get complete isoform mapping because we're looking at the full transcript. And then also we get quantitation because we don't do PCR amplification. So basically what your sample is what you're going to get as far as your data downstream. I do want to caveat one thing, and that is, while most traditional cDNA methodologies lose the poly-A tail information, Oxford Nanopore's cDNA PCR kit also will provide poly-A tail length information. So I'm also one of the designers of that, and I wanted to make sure that we were able to measure tails on our cDNA PCR. So I just wanted to make sure that most methods you lose that information, but not with Oxford Nanopore on the cDNA side of things. However, if you want modifications and no amplification biases and those sorts of things, then direct RNA is the way to go because you get all of that information in one. So just to sort of reiterate that, it's, our traditional way of looking at it is anyone, anywhere. Also you get sort of this richer insights as to what I was mentioning before, where we get native RNA data, the modifications, the tails, etc. We get fast real-time results. The prep is very quick. Once you get it on the machine, you can get your data right off the machine as it's being generated. So it's very rapid to results. And it's also quite affordable. So we have scalability within the machines that we have so that we can either look at smaller sample-based methodologies or much larger sort of high throughput and high output machines such as PromethIONs. So you can go from something like a MinION flow cell to a GridION flow cell. So a MinION flow cell to a PromethION flow cell, which gives you much more data, depending on your needs. However, I would always argue that if you're doing transcriptomics, PromethION is the way to go because more data, more better when it comes to transcriptomics, because you want to have a good depth of coverage. So I'm going to walk you through the workflow. It's quite simple, but there are a lot of sort of ways that people can maybe sort of get a bit confused or a bit sort of worried about how they interpret how these things function. So the platform itself is based on the central tenet of the Nanopore. I'm going to see if my pointer will work. There we go. We've got a nanopore that's embedded in a membrane. And then we have a motor protein that will sort of help feed whatever your molecule is through the nanopore. So you've got RNA or DNA that's being translocated through the pore. And there's also an ionic current that's flowing from one side to the other. And because of that, the ionic current gets blocked at specific characteristic base resolutions. So you can actually sort of look at what the current blockage is and then extrapolate backwards and use that to base call, because we know we put sequences so that we know what they are, and then we can kind of train the models to sort of figure out what it is that's in the pore at any given time, and then we use that to train the base caller. So if we're looking at something like DNA, it's these motor proteins will function at 400 bases a second. For RNA, it's about 100 to 125 bases a second. The thing about nanopore sequencing is that it's read length agnostic. So basically what your sample is, what you put into it is what you're going to get out of it as far as data. We can do PCR-free preps, as I mentioned before, which direct RNA sequencing is. Of course, we can sequence DNA and RNA, which we're the only platform that can sequence RNA directly, which is what I'm talking about today. So it's quite an unusual and very powerful methodology. You also get real-time analyses, as I mentioned, and then we have all of these things that we've sort of come up with and engineered over the last almost 20 years now are scalable from small to larger high throughput scalability, as I mentioned before, at low cost. So when it comes to direct RNA, the kit's been out since, so this sort of ability to sequence RNA directly has been out since 2017, is when we first released the kit. And we've had a couple of iterations of improvements, but there was a bit of a dearth of improvements for a while, particularly like throughout the pandemic, where we were working on other things. But last year, in the last 2 years, we've sort of really focused on trying to make some significant upgrades, which we have done. So last year, we released in November a new kit upgrade chemistry from what was baseline at the time, which was RNA-002 kit, which was on the on a specific chemistry that had a R9 pore, to a new chemistry, which is RNA4. That's the name of the chemistry that we talk about, and also the kit itself. So we have RNA4 chemistry, which has a new pore, which is RNA specific. It's been engineered so that it works very well for high accuracy for RNA. Whereas R9 was usable for both RNA and DNA, which is, but this particular pore is being discontinued. So now we have a new one that's much more powerful for RNA specific. We also have a new motor protein, which moves RNA, which improves the RNA output. So combined, these two, the new motor protein and the new pore, the new RP4 pore, have increased accuracy as well, which I'll go into in a bit. But we have a whole new set of chemistry, which is its own platform upgrade, and it has its own motor protein. The RNA kit does not work with any DNA preps, so you can't run cDNA preps with the RNA motor protein. It doesn't work. And also anything that's DNA will not work on the RNA flow cells. So these Min4RA or Pro4RA, those are specific for direct RNA only. But it makes it way better. And then we've also done some improvements on the software. We've improved the accuracy. And then we've also included poly-A tail length, and we've got some new modifications that we've been looking at, particularly M6A and pseudo-U, that have been included as far as standalone base colors that you can use for the data, which I'll explain in a minute. So the things to consider when you're actually starting RNA work, which I think a lot of people can get quite scared about, but it's really not that. It's not that scary. You just have to be a little bit, I guess, meticulous. The one thing that you want to be very careful about is RNA degradation. So if you're working with RNA, it is the big scary thing is whether or not you're going to introduce degradation, and that is through all the RNases that are present everywhere. So just remember, don't touch your face. Don't touch anything with dirty gloves. Clean your gloves. RNase Zap is your friend, or any of those other kind of RNase remover type of chemistries. So be very friendly with those. Make sure you wear clean gloves at all times. But there are other ways that you can avoid using or sort of avoiding degradation, and that is making sure that you have good extraction protocols, because what you want to do is you want to be able to get the longest RNA transcripts possible. Again, when you're sequencing RNA, each transcript is important, right? This isn't like DNA where you're trying to get coverage of a single single bit of information. Each one of them is important because they each represent either a gene that's been transcribed, or so you can look at the levels of transcription, whether or not this gene is highly turned on or turned off or whatever it is, or which isoforms are being used. So everything is important. You want to make sure that every isoform and every transcript is full length so that you know what it is that you're looking at. So very important. We need to be very careful on how we store it and how we handle it. But the other thing we need to be sort of focused on is extraction methods. We can introduce degradation through particular extraction methods. We at Oxford Nanopore have been working through some methodologies that we know work for us. And we have released those on the Nanopore community. So if you become a Nanopore, those who are already Nanopore users know that the community is there for sort of as a repository of information, both from Oxford Nanopore, but also from other users who love to sort of come on and talk about, I've tried this, have you tried that? This works for me. I did this and it worked out beautifully. And then also, like I said, stuff that we have tested and we support as far as protocols and things like that. So the community is available for everybody who is a Nanopore user. And if you aren't a user yet, and you will, when you become one, you'll see that there's just a ton of information there. Some of the things that you'll see are things like RNA extraction that we've worked out to try to get the longest transcripts possible and, the sort of best quality. We've also looked at like what works best as far as poly-A enrichment. So, when you extract RNA, you get total RNA from a cell, but 95% of that is ribosomal RNA or tRNAs. Some people are very interested in those. Most people want to look at protein encoding RNAs or mRNAs or messenger RNAs. And those sometimes you are better off enriching for because that's only 1 to 5% of your actual total RNA extraction. So sometimes it's nice to sort of just yank those out and look specifically at those and not worry about the 95% of other stuff, which to you would be junk, to others not so much. So we have methodologies of looking at poly-A enrichment, and that's either through enriching for poly-A specifically using something like an oligo DT pull down, or ribosomal depletion, where you go in and you select for, or basically you're either using oligo DT beads to poly-A select things that have a poly-A tail, which are usually messenger RNAs, also long non-coding RNAs, or you remove everything that is tRNAs or ribosomal RNAs. So that's ribosomal depletion. So you go in and just basically yank out all the stuff that is ribosomal or tRNA, and then you look at whatever's left. We have methodologies for both of those and protocols that we know work quite well. We also look at things like polyadenylation of non-poly-A. So if you're looking at something that doesn't have a poly-A tail and you want to be able to use the kit, which is poly-A specific, we have methodologies that have worked for us. For adding a poly-A tail. I think one of the big surprises is that most people, when they try to add a poly-A tail, will try to run this reaction to, as long as they possibly can. But actually, what we found works best is if you only run the reaction for 30 to 90 seconds instead of 30 minutes. So again, we have things that we've tested and tried and true that have worked for us, and their protocols are available. The other thing that that you want to consider if you're doing RNA work is effects of low RIN. RIN is RNA integrity number. This is a methodology where you're comparing ribosomal RNAs, both 28S and 18S, so small subunit, large subunit fragments. And you can compare these and look at the percentage of how much your sample is degraded by looking at the degradation of these two very highly abundant large transcripts. So this is something that Agilent has put out. There's some papers on this. And we measure these on the bioanalyzer, which I know is being discontinued, but there will be other ways of measuring this, where you want to look at something where if the 18S and 28S are highly, are intact and they are sort of their full length, and then you get something like a RIN of 10. It's a 1 through 10 kind of grading. Things that are more degraded, you can see the 28S is a little bit more degraded here. That's a REN of 6. The more degraded your sample, the more you lose those ribosomal representatives down to REN of 3 and REN of 2, and that means your sample is pretty horribly degraded at that point. So what we recommend is a REN of 7 and above, and we have sort of knowledge, what do we call know-hows, that are available to sort of look at the effects of what happens if you have something that's at a REN of 6 or REN of 3 compared to something where it's a nice REN of something that's 7 and above. And then we also have a non-poly A specific sequence prep. So if you have something that you know the 3' end, we give you the ability to sort of target those particular transcripts. So something like ribosomal RNA. The kit that we provide is polyadenylated specific. So something like an mRNA would be your target in that from the kit. But if you want to look at ribosomal RNA, we give you the methodologies for designing your own adapters, which then allow you to use the kit to sort of target things like ribosomal RNAs, and we're working on tRNAs as well. So now that you've sort of approached this with the ability of having the best quality RNA, what does the kit actually, how does it work? So what we've got here in this panel is the methodology. So again, I mentioned that it works on things that are polyadenylated at the 3' end. So the kit has, so this would be your mRNA. We have a reverse transcription adapter, or an RTA, which has a poly-T underhang or overhang, however you want to look at it. This will anneal to any sort of polyadenylated 3' transcript, long non-coding, or messenger, for example. This will anneal, we ligate this to the 3' end, and this allows us to then start reverse transcription downstream of the poly A tail. So we have a reverse transcription step. We say that it's optional, but I would argue that it's worth doing because it helps you with increasing the sequencing output at the end. And the reason why we have this step is because that by generating that cDNA through this reverse transcription step, it helps to remove secondary structure. So you're creating an RNA DNA hybrid. We don't fully convert it to DNA because obviously this is direct RNA sequencing. We want native RNA. But we do want to be able to sort of have the ability to remove secondary structure so that when the RNA is translocating through the pore, it's in a nice sort of more linear fashion. And you're not working, the motor protein is not fighting through secondary structures on its own. So we have here RNA DNA hybrid. We then attach our sequencing adapter that has a motor protein on there. And then we load that on the flow cell. So in this instance, we've got the motor protein here in purple, which you would see here in purple. And what we're seeing here in this animation is the RNA is being translocated through the pore. The cDNA that we've generated of that cDNA hybrid with the RNA molecule is being sort of peeled off on the cis side or above the pore. It's never read. We don't ever use that to base call. We have no information that's based off of cDNA or DNA sequencing in this particular kit. It's all the RNA that's going through, and that's generating the signal that we then use to base call. And then we can also continue to look at the modified bases that are present within that particular RNA molecule, which we would have lost in that cDNA conversion. So, and also there's obviously no PCR biases because we're not PCR amplifying it. So one of the things that we've done recently in the last couple of months is we've changed the prep a little bit by adding a new reverse transcriptase. This has shortened the prep time by 20 minutes, which is great. The kit itself is very quick to do compared to cDNA methodologies because it's only about, sort of maximum about 2 hours, depending on how many you're doing, obviously, at any given time, but it's usually about, just under 2 hours to do a prep. But by changing this reverse transcriptase, what we were able to do is we were able to capture longer reads. So we get more of that cDNA RNA hybrid, which means we get much better ability to sort of capture longer reads. So this is the old reverse transcriptase that we were using. In this panel, you can see we've got the read lengths using the old, and now with the new reverse transcriptase that we've added into the protocol, which is a third-party reagent, we now get longer transcripts, which is good, whether we're looking at poly A or total RNA. As I mentioned, The kit works on enriched samples or total RNA. So if you've got a very small amount, you can go ahead and use total without having to sort of enrich and reduce the amount of RNA that you've got. That reverse transcription adapter here will target anything that has a poly A. So even in a total RNA prep, it's going to target your mRNAs or anything that has a poly A tail. By adding these new reverse transcriptases, we can get longer transcripts. So this is a Lexogen spike-in serve mix4 panel. And these are looking at sort of longer transcripts. And we've got a 4 kb transcript, a 6 kb transcript. Again, this is read agnostic. So if your RNA is really, really in good shape, whatever you've got will go through the pore. I think the longest in-house we've ever done is about 19 kb. And that was an IVT transcript that was really hard to make, but it was possible. And then I think externally customers have done the full length coronavirus genome, which is about 27 kb, 28 kb. So it just depends on what your sample is. Again, it's length agnostic. So whatever you can get to the pore is what you're going to get out of it. The sample preps for the sample prep input amounts are 1 microgram of total or 300 nanograms of poly A enriched. So it just depends on, however you want to go about this. And then the other thing that we've done recently is we've improved the run conditions. So this is new. So the kit went out, this RNA4 kit went out in November of 2023. And we've just released these in May of 2024, these new run conditions. These are sort of changes to the software. The only changes to the prep are the new reverse transcriptase, but we have changed the software so that we can increase output by about 20%. So here we're showing total RNA preps that were done with universal human reference RNA or UHRR. Again, this is something that you can buy externally. This is from Agilent, the one that we use. And these are like 10 cancer cell lines that are put together into one really nice sample that you can then use to sort of test. We use it quite regularly to test our improvements. And here we're showing number of reads on a PromethION flow cell, the new run conditions versus baseline. And what we see is we get better pore occupancy in the new run conditions after about 14 to 20 hours is where normally we'd see a little bit more pore occupancy drop off in the old kit conditions. Now we're getting about 20% increase after that. So this is all something that, again, this is a software change. This is available in Minnow 6.0 release, which is coming out, as I mentioned, sometime in the next few months. So one of the other improvements that we've done, again, this is sort of a software improvement, is obviously Oxford Nanopore is a long read. We're known for long reads, but a lot of people who work in RNA space want to look at short read things. And there's a lot of work in RNA that's sort of on the shorter side of stuff. So people that work on tRNAs are only about 70 nucleotides. So in the past, we've always recommended that people would do RNA preps that were 200 nucleotides and above. But now we've changed the software so that we can write out really nice mappable data that is 50 nucleotides and above. So this is something that's going to go out. These data will then be generated as part of the default. So if you're running a normal length library prep, you will get this increased amount of data that's 50 nucleotides and above. So anything that was sort of between 50 and 200 are now going to be present in your output. So if you, if you have shorter samples, so people who are looking again at tRNAs, or if you've got a degraded sample, or you have a sheared RNA sample that you want to run on the Nanopore platform to get mods, with this short read enabled software, we get incredibly high output. So this is a PromethION flow cell. This is an IVT transcript or in vitro transcribed transcript that's 90 nucleotides. And we ran them on a PromethION flow cell and we get over 100 million reads. So if you're looking at something that's already short and you want high output, you will get that. So this is baseline before, when this was sort of the cutoff was 200 nucleotides and above. Now with 50 nucleotides and above, we get an enormous amount of output from these same flow cells, same kit. So no change, it's just a software thing. And we've got, here's the sample where we're looking at, this is the 90 nucleotide. It shows the read lengths here, are, quite nice. So this is something that's new. Again, this is coming out in Minnow 6.0 when that's available. So one of the things we always want to look at when we're looking at RNA being dynamic information, it's not about, X amount of coverage. It's about looking at whether or not something is, this is dynamic information. And so therefore we want to make sure that it's reproducible. So we always want to make sure we want to look at replicates when we work with RNA. We usually work at 3 or above of the same sample, whether they're biological replicates or technical replicates. We want to make sure that everything is reproducible. And we want to test that too every time we make these changes. So we tend to look at things like Lexagen's spike-ins. So we've got serve mix 3, and we look at different replicates comparing gene counts to make sure that we're getting the same representation each time that we do the prep. Again, these are using the Lexagen spike-ins, and we're looking at comparisons between 2 technical replicates, and we get 99% correlation, which is very, very, very good. So we know that we're not missing any data. And we can see that also here when we look at the replicates for the transcript lengths. So we've got replicate 1, 2, and 3, and we're looking at 2 different serve panel transcripts, and you can see that the representation is the same as far as transcript lengths. So we know that this is reliable for data generation, which is very good. We've also tested that on the human sample, the UHRR. Looking at gene counts again, we get high correlations, 98 or 99, depending it's Pearson or Spearman's. And we look at the 2 different replicates, we see high gene count correlations, which is very good. So we know that the kit does produce robust data. Also, we look at sequencing output. So again, I was mentioning we've sort of increased the output over time. We've got aligned read output for a PromethION flow cell, which is about 20 million reads, and 5 million for a GridION. Obviously, GridIONs or MinIONs have, the GridIONs run MinION flow cells. Those MinION flow cells have fewer pores, and so you get small, sort of fewer reads per output. So we'd expect that. Read lengths are good. Again, it comes down to how your RNA is extracted, but for this human representation, we get very good long reads, a median of about 1 kb, which is what you would expect with human. Read N50, which is an Oxford Nanopore sort of way of looking at things, is over 1800 nucleotide read N50, which is very good. And then when we look at transcript coverage, we see that majority of them are at least 100% as far as full length. So that is exactly what we want. The other improvements that we've made recently are we have changed. So I mentioned before that we've changed the port and the model, the motor protein and the model to sort of increase accuracy, which we went out with higher accuracy back in November. Well, of course, being Oxford Nanopore, we've changed it so that we have higher accuracy again. And so the November models that we were producing before were giving us about, for a human transcriptome, 95 to 96% accuracy using the high accuracy model, and then 96 using the super accuracy model. We've had a bit of a step back over the last 6 months and changed it again as far as the base calling accuracy, base calling architecture. And we've expanded the data sets, so we've included mod-based data as well. And so we've increased the accuracy significantly. So we've got the new model with the high accuracy model is at 97.5%. And then the super accuracy model is at 98.8%, which is very, very good for RNA. So again, this is looking at native RNA with modifications, sequencing, single read, 98.8% accuracy using the super accuracy base caller. Now, super accuracy base caller is still quite compute intensive. So if when you're running it on the machines, sometimes it's easier to do the high accuracy base caller. But if you really want to push it to get that higher accuracy at the very end, you can rebase call the data with the super accuracy model or the SUP model and get that higher accuracy using it sort of like as an offline base caller. This is available in Dorado right now. Dorado 0.70, which went out in May. This is all the same flow cell, same kit. If you've already generated data on RNA-4, say, for example, before these models were released, it is backwards compatible, so to speak. So if you've already got those data and you've got the Fast5s or the Pod5s, you can run them through these new base colors and get that accuracy again. So it just depends on, whether you've got those data. So we're very excited by this, which is, really good because we're getting close to what the DNA accuracies are, which is good. So that's kind of the nuts and bolts of direct RNA sequencing and how, sort of how the data will look when it comes off the machine, but actually what can you do with it? So one of the things that we use direct RNA sequencing for is to look at, transcriptomic information. So again, as I mentioned, we want it to be robust data. We want it to be something where you're looking at dynamic ranges because some things are expressed at very low abundancies, some are expressed at very high abundancies. We want to be able to sort of capture all of that information because it is on a dynamic scale. So we test with, again, known inputs. So known spike-ins, these are the Lexogen mix 4s again. So known spike-in concentrations of particular numbers of molecules. So we've got sort of expected versus observed, and we get very high correlations for that, which is wonderful. But what does that mean? It means that we can sort of correctly annotate gene function, whether it's up or down regulation, which then helps things like disease mechanisms, what's being turned on, what's being turned off. Again, with the replicates, we can sort of measure that and sort of look at biological replicates. How often does this happen? And, what are the dynamic ranges of what we're being able to see? And this helps people to develop drugs or targeted therapies. And we can do this with confidence because we know that, expected versus observed, we are getting high correlations between that. So we can also unravel things like the complexities of gene expression, and we can map complete isoforms. So for example, if you're doing the kit, as I mentioned, will pull out anything that has poly A tails. So you can look at long non-coding RNAs. Obviously, sometimes these are very long and therefore they're difficult to sequence with traditional or legacy cDNA methods. Whereas here they just sort of come for free. Like if they're there, you'll find them. Which is really good, and we get good representation of that. So this is a kind of a new and exciting field of people to sort of dig deeper into long non-coding RNAs. And then also for protein coding RNAs, it gives you good identification of looking at transcriptome representation. We get good numbers of transcripts. And they're also nice and full length. So we get long read, which means you can identify different isoforms comparatively. Whereas if you're trying to piece something together from short reads, it's very difficult. Isoform splicing can be very difficult to sort of reassemble from short reads. With long reads, you're basically reading from the tail to the tip, right? So because you're going 3 to 5 prime when it's going through the pore. So it starts with the poly A tail and then reads to the 5 prime end. But you get really good coverage, and you can then sort of tease out which of these isoforms is the one that you're actually looking at and find novel exons, which is really interesting. And this again helps to sort of look at drug targets. Some genes splice differently, and so therefore you need to know that if you're looking at a drug target, or for functional gene analyses, if you're trying to map a genome, you can use isoforms to try to help figure out where sort of a novel genome the representation is of what the exons are within a genome, and things like synthetic biology or evolutionary studies, which is one of my favorite things, evolutionary stuff. One of the other things, I sort of touched on this before, we get better insights of poly A tail length. So this is a particular example where we've shown poly A tail estimation. This is available in Dorado. So again, you run your samples. This information is already there. Like whether you want to use it or not, it's there. You can measure the poly A tail lengths. It gives you sort of richer insight of what's actually happening to these particular transcripts. Are the tails being shortened? because of some perturbation that you've done? Or is it because it's being transcribed, or sorry, translated or translocated within the cell? You can kind of have that kind of information. If you do a knockout, does it affect the poly A tail of other transcripts, which means that they, renders them unavailable to do proper translational processing? There's all kinds of things you can look at. This data is available. It's now available to be to sort of run through Dorado. Before, you would have to use a third-party tool to look at poly A tail lengths, which was much more computationally intensive. Now this is just available in Dorado. If you want to look at it, you can. We can look at, here we've got IVT transcripts that we've made with different poly A tail lengths up to 160, and we can measure them. We know that this is what they'd expect them to be, and that's sort of where they're mapping as far as what the estimation is. So this is quite good, very accurate. But it also means that we can look at things like the differences between poly A tail lengths of something like a long non-coding RNA versus protein coding, or also between different isoforms. Are they utilized differently? Do they polyadenylate differently because they're different isoforms of the same gene? So we can actually do, between isoforms, same gene comparisons. And this helps for looking at gene expression regulation, as I mentioned. If you do a knockout, does it affect things? And then also looking at how isoforms are used differently and looking at disease. The other thing that we get from the data that's already there, and you don't have to use it, but you can if you want to, is looking at modified bases. And so we have specific mods that we look at, which most people are very interested in. So like M6A, we had released previously a DRAC model or a DRAC motif, where these adenosines are modified within a particular motif. Now we've also got an all context model. The DRAC motif model is quite high accuracy. So, basically you run your sample on a machine, it kicks out your data, and then you can run it back through the M6A base caller, looking at the DRAC model base caller, and get you that information. You can run it again through the all context model and try to find M6As that are present. So, any kind of pod 5 or fast 5 that you've got, you can just run it through these different models. You don't have to, but if you want to, it's there. So it's a whole new way of being able to identify things that are that have implications in things like understanding cellular functions, looking at how, the differences between how things are translated, different isoforms that are being utilized, different perturbations when it comes to sort of like cancer usage for drug therapies, or even, whether or not something is metastasized and becoming cancerous versus whether it's been modified before that when it was non-cancerous. So we have the ability to sort of look at different modifications quite accurately with these different base calling models. And excuse me, also we have a native pseudouridine model. Again, this is another one that's very interesting to most RNA biologists. So this is again methylation of uridine. And so we've got pseudouridine. These are also available in Dorado. The M6A modified base colors are available in Dorado, and that's 0.70. And so you can see here, this is a ribosomal RNA where we know there are pseudouridines present, and you can see that the model calls it very nicely. So we know that we're highly accurately identifying predicted sites of pseudouridine using this model. So you run your sample, you get your information, you can base call it with the canonical base caller and get that information, which is, highly accurate. You can run it through the different modification, specific mod based colors and get that information too. So and poly A tail length. So there's a lot of information that you can get just from running one sample with one prep. So, it's incredibly rich, the amount of data that you get out of these. And then one final sort of thing that we've been working on as far as, an application for direct RNA sequencing is we're using direct RNA sequencing in a novel way to sort of do manufacturing QC for mRNA therapeutics or mRNA vaccines. And so one of the things that's really wonderful about direct RNA sequencing is that traditionally, when you're doing mRNA vaccine QC for biopharma manufacturing, you would need to use many different sequencing or many different methodologies to sort of identify different QC critical quality attributes. So what the sequence is, whether you've got intact transcripts, poly A tail length estimation, you'd have to use all these different kinds of methodologies. They're very complex. They take a lot of time. They take a large footprint, and these are highly resourced as well. With direct RNA sequencing, we can actually do a lot of these critical quality attributes in one test on one platform. And so we were looking at, if you can see here, we've got the different checks that we've already, things that we can already test for with, as far as critical quality attributes that are necessary for certified GMP QC. Identity, integrity, and purity tests are what they're looking for, and we can already do the majority of those. There are a couple that are in development, such as 5' cap detection and double-stranded RNA. So it's one test, one platform, and we also have a plasmid precursor test because these are usually made out of, these are IVTs that are made out of plasmid precursors. We have the ability to sort of put together test packs for using these for mRNA vaccine QC. One of the things that's critical for mRNA vaccine QC is the ability to train models that look at transcripts that have been 100% modified with N1-methylpseudouridine, because that's what's utilized in a lot of RNA vaccines at the moment. And so N1-methylpseudouridine, if you try to sequence that with a canonical model, will look awful because 25% of your sample is unidentifiable because it's been modified. But with direct RNA sequencing, we can train a model for that, which we have. And we had, again, an old model that we released back in November, which was at 95% accuracy, but we've improved that to about 97.2%. And we have this model that's available. This one is not available in Dorado. It's available through, the standalone Dorado that's out there. This is available through limited release and through commercial agreements with Oxford Nanopore. If you're interested, please get in touch with our business development team. And so what we're showing here is 20 in vitro synthesized transcripts that have been 100% modified with N1-methylpseudouridine, and we can see that they call them quite highly. This is all something that we've been putting together with mRNA tail length estimation for mRNA quality control as a test pack that comes with our regulated Q-line grid version 1.1, and it sort of helps to run your sample and see if it passes QC or not. So you can look at identity, you can look at poly tail length, and all sorts of other critical quality attributes all on one run, on one platform. And there's also an accompanying plasmid precursor test that's available as well. So I'm coming to the end of my talk. I would recommend that you take a look at our RNA and cDNA sequencing applications page on our website for more information about our technologies. This is available, and the sequencing kits that are available, and more related resources. The resource page is always wonderful. There's a lot of information there. There's a lot of really nice papers that we sort of discuss, stuff that we've done as well, posters and things like that. You can download the workflow there, and it gives you sort of an introduction to extraction, library prep, so on and so forth. So I would recommend looking at the resources page off of nanoporetech.com resource center, so you can see there. And just to kind of give you an overview, what's available now. So I've gone through RNA4, the improved accuracies, the improved output. We got longer transcript capture, short RNA sequencing, and more modifications. Just out of the normal data that you get from the machine, you get all this information. We are currently working on what's in R&D, our multiplexing, which I know everybody's very interested in. We are actively working on it. We don't have an estimated time for release of that, but it's sooner rather than later, right? Targeted RNA sequencing is something that we're interested in, automation as well. So we're sort of getting this onto our Elysian, as well as things that are available for third-party kind of liquid handling machines. Sample prep updates always. We're working on tRNAs at the moment. And again, the identity test for biopharma mRNA vaccine, which is what I sort of just spoke to you about 3 slides ago. So to sort of come back full circle, direct RNA is pretty wonderful because you can get things like RNA modification detection, poly A tail lengths, isoform mapping, transcript quantitation because you don't have PCR bias, gives you much richer insight, even more information than you might want, but you've got it if you want it. It's faster, you get real-time results. You can start pulling your data off the machine immediately and start looking at it and analyzing it if you're very curious, which I usually am. And it's also accessible and affordable. So it's really sort of a very powerful tool, but it's very easy to do. And I highly recommend it. And with that, I would like to say thank you for your attention. And I have had a really wonderful time discussing this, and I'm available for questions through the live Q&A right now. Thank you very much.
Thank you, Libby, once again for your presentation and for staying a little bit extra time today to answer some extra questions. For those of you who are still online, thank you for staying with us. To learn more about direct RNA nanopore sequencing, you can find more information using the link on your screen or the QR code. If you have further questions about your projects or getting started with nanopore sequencing, please reach out to your local sales support person, or you can email support@nanoporetech.com. This knowledge exchange, along with all of our previous broadcasts, are available for you to watch on demand, so please visit the Oxford Nanopore website to check them out. You can also find more information about our upcoming webinars and knowledge exchanges on the Oxford Nanopore website. Thanks to each of you, and enjoy the rest of your day.